And the topic we'll be discussing today, along with my colleague, Dr. Suman Dandamudi, is bioanalytical method validation of NDAs, what the assessor looks for. So briefly, we'll be covering what's in the new 2018 B&B guidance, and the talk will be broken up into two parts today. First, I'll cover the method validation section, and I'll also give some assessment examples and case studies we've seen in the Division of Bioequivalence. Then my colleague Suman will go through the sample analysis section of the BMV and also give some assessment examples as well as some case studies. And then we'll wrap up with some conclusions. So first I'll talk about specificity. And this is how specific is your analytical method to detect your analyte. So blank biological matrix from multiple sources should be analyzed for interference at the retention time of the analyte, and this should come from at least 6 sources. Matrix effects should be determined, and this includes either ion suppression or ion enhancement. Potential interfering substances in the biological matrix should be studied, and this includes endogenous matrix components, metabolites, and comeds. So as far as the bioequivalence assessment, what does the assessor look for? We check to make sure the chromatograms have no interfering peaks at the retention time of the analyte and the retention time of the internal standard. We also check that the comeds do not interfere with the analyte or the internal standard peak in the chromatograms. So next I'll talk about the calibration curve, or CC. So the calibration curve is the quantitation range of the assay, and it should be the concentration range expected in the in vivo bioequivalence studies. The calibration curve should be continuous and reproducible, and also it should be prepared in the same matrix as the samples in the in vivo bioequivalence study. Next I'll talk about quality control samples, or QCs, as we call them. The QC should be prepared in the same matrix as the bioequivalence study samples. We included recommendations for the QC concentrations, the lower limit of quantification, or the LLOQ, and then 3 other QCs, low QC, mid QC, and high QC. So the low QC should be about around 3 times the concentration of the LLOQ. Then there should also be a mid-QC, which is usually around the mid-range of the calibration curve, and then the high QC, which is the high range of the calibration curve. The calibration curve standards and the quality control samples should be prepared from separate stock solutions. Next, I'll talk about recovery or extraction efficiency. The recovery of the analyte and the internal standard needs to be calculated separately. The recovery of the analyte should be calculated at 3 QC levels. That's the LQC, MQC, and HQC. The recovery should be calculated by comparing the analytical results of the extracted samples with the corresponding extracts of blanks spiked with the analyte post-extraction. And this post-extraction spike represents 100% recovery. Recovery does not need to be 100%, but it should be efficient and reproducible across the 3 QC levels. So the method validation guidance does not list an acceptance criteria for percent recovery. However, the sponsors should have SOPs in place with your own acceptance criteria, and the acceptance criteria should be justified. So what does the bioequivalence assessor look for? We check to make sure that all the percent recovery values are consistent across the 3 QC concentrations, the LQC, the MQC, and the HQC. So this is where I'm leading up to the new the assessment example we've seen in one of our bioequivalence assessments. This is percent recovery data, and as you can see, it was assessed at 3 QC concentrations. In this example, they call them A, B, and C. So if you're looking at this data on the surface, it looks okay. You have 19% recovery at level A, 21% at level B, 21% at level C. So that's okay. It doesn't have to be 100%. It's consistent. But when you take a deeper dive down into the data, you can see, if we're looking at the top block for sample A, you're starting with 29% recovery during your first sample run, but by the time your run is ended, you're looking at 15.2%. So overall, your average is 19.4, but if you look at your percent CV, it's 30%, and that's extremely high for a recovery. So as I said before, there is no acceptance criteria for recovery in our guidance, but we do expect the you as the sponsors to have SOPs in place to justify any acceptance criteria you have. This should have triggered an investigation on the CRO's part before actually analyzing the samples. So on the next slide, I also have the internal standard data that coincides with the QC data for sample level A, B, and C. So as you can see, within each run, the recovery for the internal standard is also decreasing over time. So the average recovery at all three QCs was 17.99%. However, the percent CV was extremely high at 26.5%. So next I'll talk about accuracy and precision, A&P. So all A&P experiments should include a minimum of 3 independent runs conducted over several days. Freshly prepared calibrators and QCs should be used for all accuracy and precision runs. Accuracy and precision should be evaluated at the LLOQ, LQC, MQC, and HQC. So next I'll go over stability. Stability of the analyte should be determined in the biological matrix for the intended duration of the sample collection, the sample handling, and also the sample storage. Stability should be assessed for autosampler, benchtop, processed, freeze-thaw, stock solution, and long-term stability of the analyte should be determined. And this long-term stability should be determined at the storage conditions and storage temperature. For combination drug products, the stability of the analyte should be assessed in the presence of other drugs. The stability studies should cover the expected sample conditions before receipt at the analytical lab all the way up until the sample analysis. So what does the assessor look for during the bioequivalence assessment? We check to make sure the long-term storage stability is sufficient to cover the study subject sample storage times. This range of time includes from the first day of subject sample collection all the way until the last day of subject sample analysis. We check to see if the same anticoagulant was used for long-term stability and during the sample collection. We check to make sure the autosampler stability is sufficient to cover any reinjections that may have occurred during subject sample analysis. We also check to make sure that the freeze-thaw stability data is sufficient to cover any reassayed subject samples. And we also check, is the stability data sufficient to cover any processing deviations? So next I'll talk about dilution integrity. Dilution integrity is used if the subject samples have concentrations of the analyte that are above the ULOQ, then the integrity of the dilution should be validated. So QC samples above the ULOQ should be diluted with like matrix to bring the samples within the validated quantification range. Accuracy and precision of dilution QCs should be demonstrated. So what does the assessor look for during the bioequivalence assessment? We check to make sure the dilution integrity concentration will cover any subject samples that are reassayed for concentrations above ULOQ. So next I'll talk about partial validation. Partial validations evaluate modifications that are already to the already validated bioanalytical method. Excuse me. I'm going to go over some examples that are in the method validation guidance. There are also several other listed, but these are just a few. So anytime there is a method transfer between labs, there should be partial validation submitted to us. If there are any changes in the analytical methodology, as far as different type of mass spec is going to be used, that should be validated. Any changes in sample processing procedures, so if you're switching from liquid-liquid extraction to solid phase extraction, that's one example. There should be a partial method validation for that. Any changes in instruments and or software platforms that you'll be using to integrate the chromatograms, that should be validated. If there's an extension of the assay range, there should be a partial validation submitted to validate the new method at the wider range. And the new QCs should also be included at the wider range if it's extended. And also any changes in anticoagulant used for the sample collection. This does not include changes in counter ions. So if you're switching from K2 EDTA to K3 EDTA, that is acceptable. But if you're switching to heparin to K2 EDTA for your sample collection, we would like to see a partial validation to prove that this change in anticoagulant will not affect the results. And also any changes in matrices. So if you're validating in urine, but your subject samples are analyzed in plasma, you will have to submit a partial validation when you're changing the matrix. So next I'll talk about endogenous analytes. Analytes can be naturally occurring in biological matrices, including hormones or insulin, or they can come from your dietary intake. Examples of this are ions, vitamins, omega-3 fatty acids. The accuracy of the measurement of endogenous analytes poses a challenge when the assay cannot distinguish between the therapeutic agent and the endogenous counterpart. In that case, the method should be validated in the same biological matrix as the subject samples that are free from endogenous analytes, for example, strip plasma. The calibration curve should be at the expected concentrations of the subject samples. The endogenous levels of the analyte in the biological matrix should be evaluated before the QC preparation by replicate analysis. The QCs should account for the endogenous concentrations in the biological matrix, so this should be additive, and it should be representative of the expected study sample concentrations. So one of our product-specific guidances for estradiol tablets actually requires endogenous analytes analysis. So in the PSG, for estradiol tablets, the total estrone baseline corrected adjusted should be reported, and the bioequivalence is based on the 90% confidence intervals of this. So sponsors should submit estradiol unconjugated and estrone unconjugated data as supportive evidence of comparable therapeutic outcome. Supportive data should be submitted for the said analytes, the individual and mean concentrations, individual and mean pharmacokinetic parameters, and the geometric means and ratios of the means for the AUC and the Cmax. So what does the assessor look for when reviewing an endogenous analyte during a BE assessment? So we check to make sure were multiple baseline measurements obtained in the time period before the administration of the study drug. If there is a dietary intake of the compound, was there a strict control of the study subject's diet? We also make sure that the baseline correction was applied to each period. So next I'll talk about back conversion of analytes in our metabolites. Metabolites can be back converted to the analyte or vice versa prior to or during sample analysis. During the method development stage, measures should be taken to stop this process to accurately measure the pivotal analyte or metabolite. So during the method development stages, I'm talking about sample collection, sample processing and extraction, the sample storage, and the process sample in the autosampler. Instability of any analyte may also cause failure of the ISR study. So as I mentioned, the appropriate method should be taken during method development stages for these unstable analytes or metabolites, as well as the study sample collection during the in vivo BE study, so during the collection, the storage, and the analysis of the subject samples. A few of my colleagues in Division of Bioequivalence did a survey from 2007 to 2014, and the lack of these measures has been a common issue we've seen in bioequivalence, and it was seen in among 32% of BE studies in ANDAs submitted to us for review. So it's very important to take care of this back conversion during the method development before you start running your in vivo BE study samples. So what does the assessor look for during the bioequivalence assessment? We check to make sure that the validation and the subject samples were treated appropriately to prevent any back conversion, and this could be an addition of a stabilizer. We also check to make sure that the back conversion was assessed during the method validation. So I'll go through one case study, Clopidogrel bisulfate. Clopidogrel undergoes hydrolysis to form clopidogrel carboxylic acid, or CCA. So this back conversion from CCA to clopidogrel could occur in the presence of methanol, and this has been noted in the literature. So considering that the plasma levels of CCA are considerably higher than those of the parent drug, even a minimal back conversion of the metabolite will lead to a substantial overestimation of clopidogrel plasma levels, and this would bias the outcome of the bioequivalence study. So that concludes my portion for the method validation. I'm going to hand things off to my colleague, Suman, and she will go over in-study analysis.
So Leah has talked about method validation. As she mentioned, I'll be going over the in-study analysis. So for study sample analysis, what does the assessors look for? First of all, are CCs and QCs included in all the analytical runs? Does QCs cover the expected sample study concentrations? Are these QCs interspersed with the study samples not only during processing but also during the analysis? And if any case, the study sample concentrations are clustered in the narrow range of the calibration standards, then does additional QCs are added to cover the sample range? So I'll be focusing my presentation from now on these four important aspects. Those are reanalysis of the study samples, manual reintegrations, run rejections and reinjections of the study samples, incurred sample reanalysis, ISR. First, I'll start with reanalysis of the study samples. What is the documentation that has to be submitted in any case if the samples were reanalyzed? So there should be SOP, and that SOP should include the reason for the repeats and how many times these will be repeated, and what is the acceptance criteria for the final value, and if any case there needs a investigation, what are the instances that trigger these investigations? raw numerical data from both the original and the reassayed runs. If any case the samples were reanalyzed for poor chromatography, then the original and also the reassayed samples chromatograms should be submitted. And finally, in the bioanalytical report, there should be a summary table which includes the reason for reassay, the original and the reassayed values, and also the percent differences, and what was the reason for the final acceptance value. So what does the assessor look for? Firstly, are there any repeats for pharmacokinetic reasons? We usually discourage any pharmacokinetic repeats. In case of multiple analytes, if ever there is an invalid result for single analyte, did the reanalysis include both the analytes or just the analyte which has an invalid value? Did the reanalysis of CCs and QC samples perform so that you can bias the run acceptance? Because that's not acceptable. So these are the frequently reported reasons that we see for the samples that were reassayed. Analyte concentration above the upper limit of quantification. Analyte concentration is below the adjusted LLOQ in the case that the original LLOQ was failed, so all the samples below that LLOQ will be reassayed. Loss of sample during processing or extraction error, internal standard variation, and poor chromatography. So I'll go over the case study example for the sample that was reassayed for the upper limit of quantification. That is, the sample is over the upper limit of quantification. First of all, dilution integrity should be established during the method validation. And in this case, there are large number of samples that were repeated that the sample concentration is above ULOQ. In this example, the ULOQ is 500.237 nanogram per ml. And what we observed is the reassayed concentration values are significantly less than the ULOQ for at least 70% of the samples. As you can see in the example, there are 4 samples I'm showing you the example. The final reported value is 170.130, whereas the original value is 812. So there's a significant difference between the original and the final reported value. And these are like percent of the reported value to the LOQ is 34, 45. So the applicant did not do any investigation, not provide a reason why there is inconsistency in the assay results. So when they were asked to find the reason, they said they could not find any reason. So finally, the study was not acceptable because of the concern of the method reproducibility. So internal standard variation. Internal standard response is one of the important element of the run review because it not only gives the run acceptance and also for the acceptability of the study sample concentration. So what we look for, are the SOPs submitted? Does this SOP include a priori IS variability criteria for these reanalysis of the study samples? And are the IS responses consistent between the subject samples, CCs, and also the QCs? In case of isotopic IS, if there is any reason for the reassay, then are these concentration values similar between the original and the reassayed samples? In case there is an apparent trend in the IS variation or several samples are affected, did the applicants perform any investigation or did they find any root cause for this variability? So I'll go over an example. So there are 3 runs that were rejected because the CCs and QCs, they failed because of the reason of either high IS or low IS response. And the applicant's SOP criteria for run rejection is if the IS response is less than or equal to 50% or greater than or equal to 175% of the mean of non-zero internal standard areas. That is not just the CCs and QCs, the whole run. And in this case, the deuterated internal standard was used. The applicant did not submit any investigation report for having such high number of samples repeated for this reason. So when we asked about that, the applicant attributed this variability to the deterioration of the IS solution. So they bought the fresh batch of IS and made a stock solution and used that to make CCs and QCs. And then they claimed that with the use of the fresh stock IS solution, there are no runs that were rejected. However, when BOSES went for inspection, even though there were not run rejections, but there were many samples that had the highest response outside the SOP criteria. So finally, the study was rejected because the validity of the data is questionable. Without understanding the root cause for this IS variation, they just went and accepted the reassayed values. That is not acceptable. Next, poor chromatography. In any case, if there are split peaks or broad peaks or interference peaks, usually the samples will be reanalyzed. So in this case study example, there is a chromatographic interference for 50% of the runs. As you can see the snapshot of one of the sample, there is chromatographic interference and that was not resolved. So the applicant is unable to resolve this co-eluted peak based on the peak areas. So when they were asked, the applicant said, okay, instead of the peak areas, we'll use the peak heights for calculating the sample concentration. But by using the peak heights, now 5 runs have failed to meet the QC acceptance criteria. So finally, the study was not acceptable because the interference peaks do have an impact on the accuracy of the analyte concentrations. Now I'll go over the manual reintegration of the chromatograms. So what documentation usually you have to submit for the chromatograms? We need chromatograms from 20% of the serially selected subjects, and SOBs should be established priori, and it should define the criteria for reintegrations. And they should provide the reason for the manual reintegration, and both the original and reintegrated chromatograms along with the data should be submitted. So what do we look for? Is there any selective reintegration of chromatograms without acceptable justification? These acceptable justification can be such as retention time shift or co-eluting peaks or baseline noise. If there is no acceptable justification, then there should not be any selective reintegration of chromatograms, especially the CCs and QCs, just to bias the run acceptance. Run rejections or reinjections of study samples. So rejected runs. What was the reason for this rejection? Did the QCs and CCs fail the acceptance criteria? Or are there any other reasons? Like is there instrument malfunction or column leak or chromatography? Whatever the reason is, that should be provided, and also both the original and the reassay concentration should be submitted. Reinjected runs. In any case, if there is the need for the reinjection of the study samples, then was the process stability demonstrated for this entire duration of the reinjected run? This is what the assessors look for. So this is an example of a run rejections. So in this case, one of the runs were rejected for the reason of poor chromatography of the calibration standards. So the applicant went and reanalyzed these in the new run. However, the new run failed because of the QCs not meeting the acceptance criteria. Now, since there was not sufficient sample volume to do further reanalysis, the applicant stated that during the original run, only CCs have poor chromatography, but not the samples, study samples. So we'll go back and accept the original concentration. That's not acceptable. So you cannot modify the run and chromatographic acceptance criteria during sample analysis. The study was rejected and was asked to repeat. Incurred sample reanalysis. Today morning, Dr. Booth has talked about a little bit of the ISR, what to be submitted. Now I'll speak from the ANDA assessor's perspective. So conduct on at least 10% of the first 1,000 study samples and 5% of the remaining samples. Usually the sample selection should be such that it covers of entire PK/PD profile, that is the sample should be near the Cmax and also at the elimination phase. Samples should be analyzed in a different run from the original analysis. And the difference in the concentration values between the initial and ISR should be within 20% of the mean of the two values, and that too for at least 67% of the ISR results. So what else we look for? ISR values should not be used for the final pharmacokinetic analysis. In any case, if the ISR value fails, then that should not be the reason to go back and do the reanalysis of the original samples. In the same way, that should not be the reason to go back and reject the original runs too. So all ISR investigations, if ever there is a need, they should be documented and also should be guided by a pre-established SOP. With that, I conclude and go over the summary of the both validation and also in-study sample analysis, that SOP for the bioanalytical method should be established before you initiate the validation, and the validated method should always be used for the analysis of the subject samples. In any case, they need a partial validation that should be performed and submitted for modifications that was taught by Leah before my session. And if there are reanalysis, rejections, or reinjections, or any study sample analysis, they should be submitted SOP and they should be placed prior to the initiation of the study sample analysis. The reasons for repeat should always be provided along with the supporting documentation. If any case an investigation is required, it should be initiated based on the pre-established SOP, and the investigation report should always be submitted. So, challenge question. What is an acceptable range for percent recovery? Leah has talked about percent recovery during her presentation. I'll give a minute. Is it 95 to 100%? Is it 25 to 30? Is it 80 to 85? Or all of the above? Hopefully everybody got it as D, all of the above. As Leah mentioned, there is no acceptance criteria. However, you should be consistent between the all three QC samples. Next question. During the method evaluation, dilution integrity was demonstrated for a factor of 10. Which dilution factors applied to the study samples are acceptable? In any case, the samples are reanalyzed for the reason of upper limit of quantification. A6, B10, C20, D, both A and B. I'll wait for a minute. So it's D, both A and B. Anything below 10, it's acceptable. So finally, I want to conclude with the acknowledgements of our division directors, Dr. Bing Lee and Dr. Nalifer Temple. That concludes my presentation, and I will give it over to Jeff.
Thank you so much, Suman. And